Hello everyone. Hello everyone. A very good evening to all of you. Am I audible to all of you? And today we are continuing with the same unit 2, a part of cytoskeletal system and some questions from Genome Organization we will be discussing today. Okay, good evening Sunshine and Anusha. I hope so. You have studied that accessory proteins part, cytoskeletal system. Last session I mentioned regarding it. Did you all study that accessory proteins part? I mean the accessory proteins of microtubules and microfilaments. Okay. Good evening, Anchal. Yes, Nikhil, Lisa, Aishwarya. Okay, fine. So we can move on to the question. Just one question from protein targeting, which I could not take it in my last session, so I just added over here. A researcher isolated a mutant of ER resident protein, that is, protein folding enzyme, PFE, that has lost its Kedal sequence. I hope so you are aware what is a Kedal sequence is. We call it as an ER retention sequence. That means if a protein needs to enter inside the endoplasmic reticulum, it must have this sequence present. That is the address for that. So potential consequences of such a mutation are given below. So what it is mentioned, this particular enzyme has lost the Kedal sequence. So what will be the consequence of that? PFE will be found in the extracellular space, or it will be degraded in the ER, or unfolded proteins increases in the ER, and PFE will be transported to the cytosol. So according to you, the correct combination of statements will be what? The correct combination of statements will be what? Just read the question carefully. Well, question says only one thing. This PFE enzyme has lost the Kedal sequence. And you know what is a Kedal sequence. And even they have mentioned also it is an ER retention sequence. So what will be the consequence of the situation when a protein loses its signal sequence? What will be the consequence? I think this was a question came in last CSIR NET exam. As I always say you, protein targeting questions can be direct questions, statement based questions, match the following kind of questions, and also some kind of experimental questions like this. Is anyone able to answer it? What will be the consequence of a protein who has lost Kedal sequence? And the enzyme is protein folding enzyme. You might be aware we have many molecular chaperones present that induces the protein folding. We have discussed it under chloroplast, we have discussed it under mitochondria, the specific pathway. So somewhere like that, we are having a molecular chaperone present in the ER. Okay, I can see answers from Anchal, Surender, Aishwarya, and Sunshine. What about others? What about others? Anyone else would like to give your answer? Nikhil? Lisa? Bit a twist kind of question, but still you can get it. Just have a think about it. No? Okay, I got only 4 answers. So let me share the answer with you. The correct answer over here is A and C. First one is PFP will be found in the extracellular space. And Third one and the C statement will be unfolded proteins increases in the ER. So let's find out how the statement A and C are correct. See, we are aware that proteins that resides in the ER, whether it is in the luminal side or in the membrane side, must have the Skeletal sequence present towards the C terminus, which will act as a retrieval signals. Retrieval signals means by mistakenly, if that protein moves ahead, that is through ergic Golgi complex, it has to retrieve back. So for that, you are having a specific receptor present. What is the name of the receptor? What is the name of the receptor that is responsible for retrieving the protein back to the ER from Golgi? What is the name of the receptor that retrieves protein back to the ER? That is vesicle. Anchal, what you said is correct, but that is a vesicle. So it is just a transporting agent. But you have a receptor for that. What is the name of that receptor? What is the name of that receptor? Very simple. We have Kedal receptor that I had told you in my last session itself. This is the receptor which usually shuttles between the two compartments. Which compartment? Your endoplasmic reticulum as well as the Golgi compartment. Right? So, that is the via this receptor, it will be carried by the transport vesicles COF1 back to the ear. Now, it was found that if this signal, that is the Kedal sequence, is removed from a protein by some sort of genetic engineering, then what happens? The protein will be secreted out of the cell. Okay, no sequence, it can be secreted out. But on the other hand, if we have any misfolded or improperly assembled proteins that are bound by molecular chaperones, they will be retained within the ER as unfolded proteins. Do you remember any specific molecular chaperones present in the endoplasmic reticulum? Any specific molecular chaperones present in the endoplasmic reticulum? Of course, we have studied a number of molecular chaperones. Do we have any unique chaperone present in the ER? Yes, Aishwarya and Sanjan, Kedal receptor, that as I told you here. Do we have any molecular chaperone that will induce the proper folding of proteins? If you remember, in the class I had discussed about BIP proteins, that is binding proteins. And binding proteins is an example of a molecular chaperone. Okay, so that's the reason the correct combination of answer is A and C. I hope so I am clear to all of you why the answer which you had given is incorrect. Clear to all of you? Anchal, Surinder, Aishwarya, Sunshine. I can proceed with the next question. Yes? Okay, fine. Now, this is a different question. You may place it any part of your syllabus. These are some following statements that is made regarding the role of protein modifications. Of course, protein modifications we can also study in protein targeting also, maybe some part in unit 3 also, that is histone modification, like that. So, what is the statements provided over here? Addition of acetyl group to the N-terminus of proteins makes it more resistant to degradation. Attachment of hydroxyl groups to the proline residues stabilizes the fibers of newly synthesized collagen. Then glycosylation we have studied. Glycosylation makes the protein more hydrophilic, enabling protein to protein interaction or does it makes more hydrophobic, enabling protein folding? What should be the correct combination over here? The correct combination of statements comes under protein modification. Just have a think. Even I have also mentioned about the importance of acetyl group in histone proteins also. Okay, that is the part of genome organization. Glycosylation is a part of your endoplasmic reticulum. And proline residues collagen formation, again this part can be taken into consideration for the structure of collagen. Okay, Mazzy Films has given the answer as option 1. What about others? Kiran, 1. Okay. Anyone else? Just you have to think a bit. As I gave you some hints regarding acetylation of DNA, what does it do? Somewhere related to that only. Similarly, glycosylation, which we had discussed under endoplasmic reticulum, what does it do? Then this is with respect to either you can take it as collagen coming under biochemistry or the collagen coming under the extracellular matrix. I said it will be related. Okay, somewhere related. Okay, Aishwarya, Karen, Sunshine. Anyone else? Anyone else? Anusha? Anchal? Would you like to give the answers? Okay, you're going with B. Okay, fine. B, C, or D. Okay. Anyone else? See, what happens here is bit a twisty question, but yes, once you understand, things will be more easier. The correct answer over here is A, B, C. Now, why did I said statements A, B, C is correct? One thing is that either of them has to be correct, right? Because it is not possible that it will form both hydrophobic and hydrophilic together when it comes to sugar protein linkage. So either of them has to be correct. So either it has to be ABC or ABD. So the correct answer over here is ABC. Now let's check it out how. So we are aware of acetylation. Are you aware of acetylation with respect to DNA? Anyone remembers what does acetylation means with respect to DNA formation? Condensation occurs, relaxation occurs. What does it occurs? No problem, Anusha. You will be clear now. Do you remember when acetyl groups are added to our DNA? What does it happens? Histone proteins to DNA interaction. What happens? Will it be reduced or it will be enhanced? What it should be? This also I have told you in the class, the importance of acetyl group within DNA histone interaction. Will it yes, it will carry out gene transcription, because when you are adding acetyl group to a protein, what is the major function? It will be neutralizing the charge. Right? Yes. That's the reason we call it as an enhanced gene transcription. Okay, so acetylation can affect the structure, stability, function of proteins, also regulate protein to protein interactions and cellular localization. So why or how it is protecting from degradation? Because as I told you, neutralize the positive charge present on the amino terminus. So if positive charge is neutralized, what happens? It becomes less susceptible to cleavage by corresponding proteases. Apart from that, this also leads to protein interaction that will help in stabilizing the protein at the same time, as I told you, protection. Okay. Similarly, the same concept is applied to histone DNA interaction, because when the histone is, when the DNA histone combination, when this acetyl group is added, histone is having lysine and arginine residues which are positively charged amino acids. So the positively charged amino acids will be neutralized. So if that becomes neutralized, it won't be able to interact with the negatively charged DNA. So it becomes less condensed and thereby leading to gene transcription. So somewhere the concept is similar here also. He's just removing the positive charge from the N terminus. Okay, that is the first point. Next, coming to the second point, collagen is basically made up of 3 polypeptide chains which is having a triple helical structure. So somewhere it looks like this. Okay, and it is having a conserved amino acid repeats known as glycine XY. So where X could be either proline or lysine, and Y will be hydroxylated proline or lysine. Okay, either of this. If it is a proline, hydroxyproline. If it is a lysine, it will be hydroxylysine. So this hydroxylation is carried out by the enzyme proline hydroxylase, which is an important step in folding and assembly of collagen protein. So what does it do? This hydroxylation will help in formation of hydrogen bonds. And this hydrogen bonding will stabilize the triple helical structure of collagen. And this provides the structural information for both proper folding as well as assembly of collagen within the extracellular matrix. Okay, so that is the correct answer over here. Yes, everyone has mentioned as regulated transcription. Next, what we had discussed, glycosylation. Glycosylations are responsible for making the protein more hydrophilic, thereby facilitating protein to protein interaction. Why? Because by nature sugars are polar and they have hydrophilic properties, so obviously they interact with water. So when these glycans are attached to a protein, they will generate a hydrated shell around the protein. So that will help it more soluble in the environment, both inside and outside the cell. So this solubility helps in protein interaction with other molecules, including protein itself. That's the reason statements A, B, and C are correct combination. I hope so you got it. Of course, the statements we cannot see directly from two. It has come from different units, we can say. Like collagen, as I told you, it is related to biochemistry and ECM. But N-linked glycosylation, somewhere it is related to protein targeting. But again, your acetylation, you can connect it with the Genome Organization. Right? So, just a combination. Very rarely such questions will ask. Now, next question. Now, this is related to your cytoskeletal system. An investigator expresses a GFP fused protein that localizes to the outer membrane of Golgi apparatus. Upon visualizing this GFP signal in the fluorescence microscope, it was noted that this GFP is pericentrosomal in its location. And treatment of such GFP expressing cells with a newly identified drug disrupted the Golgi into small vesicles. See, this is the location. After the treatment with drug, it has degraded. Now you have to identify the list of targets for the drug. Is it dynin complex, myosin, microtubules, dicer? The correct combination you have to identify. try to bring some related proteins together. And who is responsible for organelle relocalization? Then you will get the answer. Okay, Anchal gave the answer. The hint over here is you have to think among the four targets which is being mentioned, which are correlated with each other. If so, how they are related to Golgi compartments. If you connect these three things together, you'll get the answer. Okay, Surender, Anchal, Mazi has given the answer. Others try on, you will definitely get the answer. This is how you need to learn how to approach a question. Suddenly looking into the image, many a times what happens, we often don't feel like answering it, thinking that it is a tough question. But you should not do that. Read the question thoroughly, what it is given and how you can derive answer for that. At least try once. You will definitely get the answer. Who is responsible for organelle relocalization? Like organelle localization within the cell. You have three cytoskeletal systems, proteins, right? You are having microfilaments, microtubules, and intermediate filaments. So among them, which is responsible for localization of organelles within the cell? Who is responsible for that? Absolutely correct. We have microtubules. So in that case, somewhere it is related to microtubules. So how you can combine with other proteins? Myosin can be say, is myosin belongs to microtubules? Is a myosin a motor protein related to microtubules? Is it so? Is myosin is related to microtubules protein, motor protein? No, right? Then coming to the dicer. Now this is a part of RNA interference, which is a far apart from the corresponding question. So what is left? Dynin and microtubules. So you have got A and C. So dynin complex, can anyone tell me dynin is plus N directed motor protein or minus N directed motor protein? Dynin is plus end directed motor protein or minus end directed motor protein. Okay, fine. Absolutely correct, Suresh. So basically, dynin is a minus end directed microtubule motor protein. So any disruption in the dynin function can lead to pericentrosomal localization of GFP fused protein. Then, as I told you, microtubules are involved in maintaining Golgi structure as well as positioning the cell. So that's why any disruption of microtubules will definitely affect the Golgi fragmentation and reposition during the center towards the centrosome. Okay, so that's the reason A and C are correct. Yes. Next question. In an attempt to study the transport of secretory vesicles that contains insulin along microtubules is in cultured pancreatic cells, how would the treatment with colcimate will affect the transport of the vesicles? Which vesicles? Secretory vesicles. It induces polymerization of microtubules, or polymerization is inhibited by colcimate. It inhibits vesicular trafficking through the inactivation of V snare protein, or it activates the T snare protein that helps in the vesicular transport. What should be the correct answer here? To attempt this question, you must be aware of the mode of action of colcimate. Does it causes polymerization or does it causes depolymerization? Not sure? No problem. We'll let you know now. Others? Anyone trying to give the others trying the answers? Okay, Anupam, Anusha, Suresh, Surendra, Sunshine, and Mazzy. Anyone else would like to give the answer? Okay, Anit, Anchal, and Sunshine. Fine. Let's check out the answer now. The correct answer, as told by every one of you, it is second. So, what is Colismid? It is a drug that is responsible for inhibiting your microtubule polymerization. How? By binding to the tubulin subunits. You are aware that tubulin subunits is made up of alpha and beta, right? Thus, preventing their assembly into microtubules. So, as a result, what happens? Microtubule network will be disrupted, depolymerization occurs. So, in the context of studying the transport of secretory vesicles that contains insulin along the microtubules in cultured cells, treatment with colismid is going to inhibit the transport. As I told you, if you know the consequence of colismid, you can easily get the answer. So, since microtubules serve as tracks for intracellular transport, the depolymerization result in disruption of secretory vesicles. And this disruption would hinder the ability of vesicles to go and bind to the plasma membrane so that it will start secreting insulin. Okay, so depolymerization taken place, so it is unable to migrate, unable to fuse with the membrane, thus insulin secretion is prevented. Yes. So here are some list of actin chemical inhibitors and microtubules inhibitors. You can see that some of them are depolymerizing and some of them are stabilizing. For example, lantriculate, it depolymerizes the by binding to the actin subunits. Cytoskeletal B, it also depolymerizes. How? By providing cap to the plus end. Phalloidine, it stabilizes, that is binding along the filaments. Now, when it comes to microtubules, the very common thing is Taxol or paclitaxel, which stabilizes the filaments. Nocadazole and colchicine both are responsible for inhibition. Do you remember vincristine? It belongs to which category? Microtubules or actin? Vincristine drugs. Will it be inhibiting actin filaments or microtubule filament? Microtubules. Vincristine. It is basically an anti-cancer drug also, if you remember. Yeah. Yes, that is also targeting your microtubules. Specifically, as I told you, it's acting as an, in fact, this Taxol also acts as an anti-cancer drug. Okay. Yes, Srivani. Yes. Now, this is a very interesting question, as I told you, with respect to accessory proteins and function. So, here you have provided with microtubule and actin. You have to identify the correct combination from column B and column C. Find it out. You must be able to filter out which protein comes under microtubules and which protein comes under microfilaments. Catamine, tropomodulin, tau, gelsolin. The very first thing you should do is filter out the proteins coming under whether microtubules or actin. Second thing is you have to identify the function. Once you filter out the proteins, 50% you got the answer. Filament cross-linking. Severs the filaments and binds to plus end. Prevents assembly and disassembly at minus end. Only severs filament. Okay, I can see Anit, Aishwarya, Surender, Suresh have given the answers. Others would like to give your answers. You can give the answer, everyone. Karen, I hope so. You got the previous answer explanation clearly since you were unable to answer. Saujanya, Anupam, okay. Okay, fine, Karen. You can see miscellaneous answers coming 3 and 2, 3 and 2. They do affect normal cells. There are chances. Still, people are working on that on some way. If they will be able to target exclusively the cancer cells, something like that, they are still working for that. Yes, Mazzy and Anupam. Okay, let me give you the answers now. How does it works? Check it out. The correct answer over here is 3. Now, how does the answer 3 comes? Let's check out individual points. X3A. X is for microtubule. Belongs to the category of tau protein. Tau belongs to microtubule. And tau, if you remember in the class also, I had mentioned that it's a cross-linking protein. A category of proteins which forms filament bundling, cross-linking and attachment to the membrane. So tau is one of them. X1D, microtubule, catenine, severs the filament. Severs the filament in the sense? Severs the filament in the sense what? What do you understand by severs the filament? Severs the filament means what? Degradation or breakdown. Right? Next, you are having Y2C. That means now we have come to actin. So, actin is having tropomodulin. It prevents both assembly and disassembly from the minus end. Yes. And the last one is Y4B. That is actin, gelsolin. Again, severs the filaments and binds to the plus end. So, catenine only severs the filament, but gelsolin severs plus binding towards the plus end. Okay. Now, let's check it out. What was given in the book? See, these were the examples of the accessory proteins of microtubule. First one is gamma Turk, gamma tubulin ring complex. Always remember the protein where it binds to in the sense whether it is binding to the minus end or towards the plus end. You have to focus on this. And what does it do? So, centrosome, as we are aware that it is otherwise known as mTOC. that is microtubule organizing center. So this particular ring complex will go and bind to the minus N, helps in the assembly, remains associated with the minus N. Okay, that is the first protein. Stathmin protein, what it will do? It will go and bind to the microtubule subunits and prevents the assembly. So this event is known as catastrophic event or rescue event. This event where preventing the assembly takes place is known as catastrophic event or rescue event. I hope so you are aware catastrophe and rescue with regard to cytoskeletal protein, that is your microtubules. Do you remember? Anyone remembers? Catastrophe and rescue event. What do you mean by that? So basically it is related to assembly and disassembly. So which term is specific for disassembly? Here they are causing disassembly, right? Prevents the assembly. So that means it is causing degradation, disassembly. So whether it is catastrophic event or a rescue event. Got only one answer. What about others? Yes. It has to be catastrophic event. See, remember C to D. Catastrophe to degradation. Rescue means it is surviving. That means helps in the assembly formation. So if you remember one part, the other part you will be automatically remembered. So you can use this code word CD, catastrophe, degradation or disassembly. Okay. Similarly, you are having plus tips. They are responsible for attaching to any membranous structure via their plus end. Okay. Next, kinesin 13. It induces catastrophe and disassembly C towards the plus end. And the same thing if you see XMAP 215, they are carrying out the assembly towards the plus end. So kinesin and XMAP 215 are antagonistic to each other. One is depolymerizing it, another is providing assembly or stabilizing the microtubule. Catamine, it severs the microtubule, that is breakdown. Then MAPs. MAPs are responsible for what? Microtubule associated proteins. So they will be bind throughout the microtubules, providing stability to them. Then we have a combination of filament bundling, cross-linking and other binding proteins. Other binding proteins means one cytoskeleton connected to another cytoskeleton. So in that case, we are having tau, MAP2 and plectin. So plectin is acting as a bridge that connects two cytoskeletal systems together. That is one on microtubules and the other to the intermediate filaments, or one on microfilaments and the other to the intermediate filaments, like that. So basically plectin acts as a bridge. Okay. So these are the examples of your microtubule associated proteins. Now we have actin cytoskeletal proteins. The first protein is formin. It helps in the nucleation and remains attached with the plus end. ARP2 bar 3 complex. It helps in the nucleation, but they will form web-like structure, or we can say branches. But in which end? Minus end. This is plus end. Very minor differences. Thymosin, it again prevents assembly. That means inducing catastrophic events. Profiling, it is responsible for binding the subunit, thereby elongating. You might be aware that filamentous actin, globular actin will be converted to filamentous actin. That is the active form of microfilaments. So that exchange requires ATP hydrolysis. That ATP hydrolysis is done by this profiling. That exchange is being carried out by this profiling. Okay. Next, we have tropomodulin. Tropomodulin, it prevents assembly and disassembly at the minus end. Coffulin, it binds to the ADP actin accelerating disassembly. So these two are opposite. Tropomodulin and coffulin both are acting on the minus end. But what does it do? It prevents assembly and disassembly at minus end. Okay, both are same only. They also accelerate disassembly. So tropomodulin and coffulin both are carrying out the same function, acting on the minus part of the microfilament, thereby carrying out disassembly. Okay. This tropomyosin is similar to your MAP, microtubule associated protein, which is responsible for stabilizing the filament. Gelsolin is similar to your catamine protein, which is responsible for breakdown, but at the same time binds to the plus end. But catamine does not have specific, like it will bind to minus or plus end. But in this case, it binds to the plus end. Capping proteins that will be present in the plus end prevents assembly and disassembly at the plus end. So this is two are acting on minus end. This one is acting on the plus end. And similar to your microtubules, you are also having alpha actinin, fimbrine, filamine, spectrin, and ERM. All these are coming under the category of filament bundling, cross-linking, and attachment to the membranes. I hope so you got the answer, why the answer was correct here, right? And remember, this is also a question came in the last exam. Even nowadays, what happens, in the CSIR exam, basically the accessory protein related questions are asked very frequently. In fact, every question paper you take it out, you'll find one 4-mark question being asked from this accessory proteins. So either you remember it as in the form of images, the way it is shown over here, or remember it in the form of a tabular form. That means you can create the table and try to compare and remember. That means microtubules, functions, and microfilaments. If you make this column, you can remember. Okay. So this is important from examination point of view. Next, what we are having. Just a reminder for Watson Junoon scholarship, which is going to end by August 31st. So whoever is willing to apply, please apply first. So what all programs you will get under this scholarship? You can get 1-year coaching. You can get 12 months of access to all the workshops being conducted by Biotechnica. Then we are also having many certification courses. So depending on your interest, you can choose 4 certification courses, which will add a weight to your profile. Then there will be one VIP pass to our CRISPR conference, Congress 2023, which is going to be held in the near future. Okay. Next question to you all. When cells enter mitosis, their existing array of cytoplasmic microtubules has to be rapidly broken down and replaced with mitotic spindle that is helping for pulling the chromosome into the daughter cells. So we have got the enzyme catamine being activated during the onset of mitosis and chops the microtubules into short pieces. So what will be the possible fate of microtubule fragments created by catamine? Is it depolymerization, aggregation, degradation, or translocation? What does it do? What does it do? Just now we had discussed with respect to accessory proteins. So what should be your answer? What should be the answer? There will be many confusion going on. One thing I'm pretty sure you understood what is the function of catenate because it is there in your question itself. So obviously, point number 4 and 2, no one is going to give. Because see, during the onset of mitosis and chops the microtubules into short pieces. That's what they have mentioned. So in that case, what does it do? Is it causing depolymerization or degradation? depolymerization or degradation? What should be the answer? And here we are asking with respect to cytoskeletal system, then which one should be the most appropriate answer? Which one has to be the most appropriate answer among depolymerization and degradation? Now I can see the answers being swiped to 1. Earlier you were saying answer as 3, but now suddenly everyone is saying 1. The thing is that you have to answer according to the requirement of the question. Obviously, when catamine comes, it breaks down the microtubules into short pieces. But this results in what? In terms of cytoskeletal system. Because whenever we speak of cytoskeletal system, we always speak either depolymerization or polymerization. You got it? So that's the reason your answer should be depolymerization. So whenever you answer any question, read the question thoroughly, then give the answer. Of course, there are chances that when we look this question, ma'am has told catamine is responsible for degradation. Of course, it is doing that, but the more precise point will be if you see it as a depolymerization. Polymerization and depolymerization are the processes being carried out by our cytoskeletal system. So that's the reason we have to opt for depolymerization. Okay, I know this was an easy question, but still, why I had shared this question in order to differentiate these two terms. So, whenever you come across any such queries, think twice and give the answer. And this was, I think, a 4 marks question, I guess. I think 2 years back, somewhere they have asked this question. Okay. Next question. Cellular actin levels can be as high as 100 to 400 micromolar. Of this, unpolymerized actin concentration will be 50 to 200. Critical concentration for actin polymerization in vitro will be zero point 0.2 micromolar. So some of the following proteins are given that can inhibit the polymerization of actin in the cells. So you got the options right. I mean the thymosin beta 4, capping protein, tropomodulin, and XMAP 250. You have combination this. Which of the following options list all the inhibitors? You have to identify that. ABC, BCD, CDA, A and B. You want to see the previous slide? You can remember this, right? Like you can say ABC, BCD, CDA, and A and B. So better I can place this slide so that you'll get an answer. The options here, I'm just noting it down. ABC, BCD, CDA, and last one, I guess, A and B. What should be the answer over here? Read the question thoroughly and give the answer. Just now we have discussed all the inhibitors, and you also said you have studied inhibitors in the sense the polymerization proteins and all, microtubules and microfilaments. Just now we had discussed, plus you have also studied. So find out from there only you have to get the answer. Tropomodulin is inhibiting the polymerization. Okay. Another thing that I want to add it over here is, as always I say, read the question thoroughly. What exactly they are asking. Then you can have a clarity on regarding which answer should be a correct. Are you able to understand what I'm trying to say, everyone? Here, some actin levels are given. Polymerized and unpolymerized actins are provided in vitro condition. You have to identify the proteins that is considered to be inhibiting the polymerization of actin filaments. What should be the answer? What should be the answer? I'm still getting miscellaneous answers. 314. Find it out. You know it. Just read the question thoroughly. You will get the answer. Everyone, try it out. Anusha, Anchal, Shrivani, Sunshine. Is it that much tough? Okay, let me give you the answers. No one else is giving the answers, right? Okay, let me give you the answer. And the correct answer over here is answer 1. I want to listen the justification why this answer 1 ABC statements are correct and why XMAP to 15 is incorrect. Can anyone tell me? Can anyone tell me why D is incorrect and why ABC is correct? Can anyone give me the correct answer over here? Surinder and Anupam would like to give the answer. Absolutely correct. See, why I was keeping on saying you that read the question properly, read the question properly. The thing is that, see, here they are asking you the inhibitors of actin filaments. And the numbers they had provided you is just to scare you, because many a times life science students, when they see the numbers, they are afraid to give the answer. But that you should take it as a challenge, because there is nothing related to numbers they are asking over here, and there is no majority of the cases from unit 2 you will not expect any kind of this calculative questions. That is very rare and exceptional case. Okay, so here they are just asking you the protein inhibitors. But as mentioned by Surender, yes, you are correct. This is an example of microtubule proteins, which is responsible for binding to the plus ends, thereby providing stability. But remaining all the three belongs to microfilaments, and we are speaking about the actin filaments here. I hope so you got it. Those of you who has given the incorrect answer. Did you get it? We are asking about the microfilaments inhibitors. We are not asking about the microtubules inhibitor, and XMAP is coming under microtubules. So easily we can eliminate that, and ABC will be the correct answer. Okay, so this is yet another example where I told you to read the question thoroughly. Okay, okay, then I hope so everyone has got it. Why answer 1 is correct. Okay, fine answer. Okay, Anupam. Okay, Shrivani. So, when bound to the actin, thymosin beta will inhibit the exchange of nucleotide bound to actin by blocking its dissociation. Same is the case with capping protein. If you remember, when we speak of microtubules, we always say as plus end and minus end. But when we speak of microfilaments, we say barbed ends for plus and pointed ends for minus. Tropomoduli, They are cytoskeletal protein which inhibits tropomyosin binding to the actin, somewhere inhibitor only. But XMAP is a microtubule plus N-derived binding protein. So we can easily eliminate that. Okay. Next question. Okay, this much about the cytoskeletal system. The next question over here is Genome Organization. Which one of the following statements is true about human chromosomes? Chromosomes have highest gene density generally localized towards the center of the nucleus. Chromosomes will have highest gene density localized near the nuclear periphery. Centromeres of different chromosomes tend to cluster together at the center of the nucleus. Chromosome positioning in the nucleus is absolutely random. So you have to identify the correct statement from here. Identify the correct statement regarding human chromosomes. And basically from Genome Organization, you always get a direct question. But the thing is that you have to be very clear with the genome organization, how nucleosomes are formed, how the histone proteins are interacting with DNA, how the DNA wrapping takes place, what are the proteins involved, why it is happening, when it will be in the less condensed state, when it will be in the highly condensed state. So these are some basic concepts which you have to remember. If you remember that part, it is easy for you to attend any questions from Genome Organization. Yes, can I get an answer from you? Which of the following statements is correct with respect to human chromosomes? They are asking you the true statement. Sometimes what happens, they will ask you regarding the incorrect statement. So you have to again read the question thoroughly. Then only you can identify whether they are asking you a correct statement or an incorrect statement. Okay, Vani Shri, Anupam, Srivani, Surender and Kiran. Mazzy. If you get an answer of this, you must also justify why it is so. See, one hint which I can give you is that here they have provided you a statement. Chromosomes with highest gene density are localized towards the center of the nucleus. And here they have mentioned that localized near the nuclear periphery. So obviously anyone can guess one thing. Either of these two statements would be correct. So you can easily eliminate option 3 and option 4, because it is not possible that these two statements will be correct together or false together, but one of them has to be correct. Either it should be present in the center of the nucleus or it has to be present in the nuclear periphery. Not sure with density dependent localization. Okay, no problem. No problem. I will explain you after this. So what should be the answer? I can see now a lot of answer 2 is coming. Okay, let me explain you this. The correct answer is first. See, the positioning of chromosomes within the nucleus is not arbitrary. That means the positioning is not random. It is influenced by a variety of factors that contributes to gene expression, transcription, and overall nuclear function. Why? Because replication and transcription takes place where? Within the nucleus. That's why those factors will be contributing to the position. So we can say that nucleus has a radial organization wherein chromosomes with highest gene density are preferentially disposed towards the nuclear interior, whereas the gene poor chromosomes will be located towards the periphery. And another point to be noted here, what? This organization is conserved in all the vertebrates. Okay, suggesting that nuclear interior will facilitate or create a permissive environment to carry out the gene expression. Next point, while centrosomes play a role in chromosome organization, they will not necessarily cluster together at the center of the nucleus. In fact, you might be aware the function of centrosomes at different stages of cell cycle. So depending on that, it will be undergoing the change. So that's the reason statement 1 is considered to be the correct answer. I hope so everyone has got it. Everyone understood it? Okay, remaining questions from Genome Organization we will be discussing in the next class. And provided that I will be taking questions from cell signaling 4B. So kindly study that portion and also come to the session. Once I am done with Genome Organization, we'll move to cell signaling. If possible, next session itself. Okay. Thank you so much, everyone. See you again in another session.